All right, let's do a quick translation video. I'll try to make it quicker. Let me close me off in case I block the slides. All right, we've already learned our central dogma that you've got. Oops, let's turn this on. You've got a big, huge bunch, thousands of base pairs along DNA. And you guys have 23 pairs of them. 5' to 3', 3'. And anytime you have to make more DNA, that's replication, DNA replication, right? Slightly more complex, at least as you guys are learning it, than transcription, which is when you're finally going to take some piece of that DNA, segment of the DNA, there's going to be some promoter sequence to start transcription and a termination sequence to end transcription. And at the end, you're going to have just a single-stranded mRNA transcript. So this is called transcription. Transcription goes from DNA to RNA. Okay, and so now you have this specifically mRNA molecule, and you put some tags on it, poly A tail, you put a 5' cap, and somewhere along this little region to protect your mRNA is very precious, and the world outside of the nucleus is harsh and dreadful. And in here you have a bunch of the information that was just copied from that template strand of DNA in RNA form, right? So you're going to have U's and G's, and somewhere along here you're going to have an AUG, which is your start codon. But right now we're still in mRNA. We're still in the nucleotide. Translation is finally we're going to get to take the information that's in here and use it to piece together a sequence of amino acids. Okay, and that's translation. You're going to translate the code in the codons, nucleotide form, finally to amino acid sequences. Amino acids put together. Remember, that's your polypeptide. And that's going to needs mRNA. And it needs your ribosomes, which happen to include our RNA. You can think of them as being our RNA. And then a transfer RNA molecule. All types of RNA, single-stranded, uracil, thymine. And because it's single-stranded, these guys fold into different things, in different configurations, have different functions. But together, these guys are going to help make that polypeptide. All right, so let's look at translation. Finally, to remember to do translation, just in case you guys didn't get a chance to review. What we're going to do is we're going to use our genetic code over here. We're going to use the codons on mRNA to figure out which amino acid, and here are all the amino acids they're going to be strung together by that ribosome. Okay, so let's review how that works. If the codon on mRNA, I cannot emphasize this enough, it has to be on the mRNA. It says AUG. You look for the first base over here, the A, the second base at the top. It's the second base at the top over here. AU, and then you look for the G. You can look for the third base here, or you can just sort of scroll down on that book. This is methionine. Every start translation, every beginning polypeptide is going to start with methionine. CAU, I'm just going to do a couple of them. So here's C. Look for the second base down this way, and then I'm just going to look down. CAU stands for the amino acid histidine. And so the codon means it's on the mRNA. It's the 3 nucleotide sequences on the mRNA. Every 3 nucleotides on mRNA, remember, equal 1 codon. And 1 codon, as it says down here, like AUG, is going to tell me, gives the ribosome the instructions, place, let's do GCA, G, second base C, and then A. For this one, for GCA, it says place alanine. You can just call it ALA, alanine here or next. Okay. Practice your I'll do a little practice read. All right, so let's go up. Sorry, I'm gonna make you guys dizzy. First step in translation. Let's talk about, it says you need 2 important steps. You're going to need to be able to match a tRNA's anticodon. Oops, that's this one over here. You're going to have to match the tRNA's anticodon to the mRNA codon. In other words, you're going to use some base pairing rules will apply to match the tRNA to the mRNA, to the appropriate, to the correct mRNA. We jumped around. First, there's going to be a special enzyme, don't worry too much about it, that's going to attach the correct amino acid to the correct tRNA. And it happened in your little figure. It already happened over here. So some enzyme, don't worry about the enzyme, it's like amino T synthase, something like that, attached the proper amino acid, AA, to the tRNA. So in translation, all of our RNA, all of our favorite RNAs are going to come together. You're going to have your rRNA in your ribosomes. Notice right here, you have 2 units, a small RNA or a small ribosome part and a large RNA part. So you've got your mRNA and you finally have these tRNAs coming here. tRNA coming with the correct amino acid. And all together, they're finally going to use the instructions on the mRNA and bring the correct amino acids to that location on the ribosome on the mRNA until you're finally stringing together that polypeptide. This is how you take A, T, C's, and G's and eventually get them to, what is it, trypt, phenylalanine, glycine. Okay, so a little on your tRNAs. Yeah, I'm recording right now. Good. tRNAs. So a tRNA, again, it's an RNA. It stands for transfer RNA because it's transferring an amino acid to the correct mRNA codon. But I like to think about it as tRNA because it kind of looks like a T, right? And it folds itself into this funny T-looking shape. And at the bottom right here, you have this 3 nucleotide RNA nucleotide sequence called the anticodon. Anticodon because it can base pair to the codon. Okay, so yes, that means that the AAG that's over here, it can base pair to a UC on an mRNA molecule. Okay, but remember, when you're reading off the genetic code, you better read it off from the mRNA, not from the anticodon, from the mRNA. Although you can figure out what the codon is, right? All right, so anticodon can base pair with complementary mRNA. Ribosomes. Ribosomes, I already told you. Ribosomes are the big factory in which all of this is sort of housed and happening. But they also have, they're made up of lots of proteins and lots of rRNA. Okay. The rRNA is probably what's acting like the actual enzyme that's going to attach amino acids together. I'll show you right now. All right, let's look at your ribosome. On your slides, I believe I put an extra little picture. I put this picture. And I made it nice and big looking for you guys so that you can see the 3 different sites. You've got a small, let's point them out here. You've got a small subunit that's going to be the first thing that's going to attach to the mRNA. Once that's attached and the first tRNA is in, the large subunit is going to come in. But then notice there are these 3 sites in the large part of the ribosome. The E site, that P site, and the A site. I call it the EPA site. A is where the amino acids begin to bind. It except for one, the very beginning one, of course. P is where the peptide is being elongated. You can go up here and you can check. P is with the tRNA that's going to be holding the growing polypeptide chain where the protein's growing, or polypeptide is growing. Growing here. And the E site is going to be the exit site. This is going to be where the tRNAs that aren't being used at that moment are going to be popped off, as you can see over here, right? Exit site. This is a nice pretty picture of what it looks like in a 3D model. EPA site, E being the exit site because the mRNA is moving like in that direction. Yay, 3 stages. We've seen these names at least: initiation, elongation, termination. You're also going to have all sorts of other proteins that are going to help you in this process, but your main guys are going to be your ribosomes, right? Your rRNA, your tRNA, and your mRNA. But of course, we always skip that you have more helpers more protein helpers than just that. All right, let's start with your initiation stage. Let's see if I have a picture of these for you guys. If it's in the next slide. Is it? Well, yeah, but I can do sort of like a little drawing here for you guys before I show you the fancy drawing that's on in your book. All right. So the first thing that's going to happen, let's draw myself an mRNA, and let's say that it's, I don't know, pink. It's my mRNA. And it, it's already processed and it left the nucleus and it's got its poly A tail and its 5' cap. The first thing that's going to happen is that there's going to be a small ribosomal subunit. Should I make it pink? No, let's make it brown. Like this. And it's going to attach. And it's likely attaching somewhere near this 5' cap and the untranslated region that comes right before the first AUG, 5' cap. These guys help bind to ribosomes. Previous lecture or video, if you looked at the video. So there's your small RNA. I'm going to call it small RNA, so you remember it's your ribosomes have RNA. So it's going to bind to the small RNA, and it's not really telling me here, but it's actually sort of moving along until it finds the first AUG. Oh, I really should have drawn this better, bigger, but hopefully this will work out, right? Because that's going to allow, only then will that allow the proper tRNA to come and base pair to the AUG. The proper tRNA, the special initiator tRNA, the proper one, the correct one, has an anticodon that can base pair to the AUG. So in this case, it's got to have a UAC, okay? And then if you look at your genetic code and you read off, but you also notice the very first step in translation, if you read off AUG, you know which amino acid this tRNA is holding. It is holding the amino acid methionine. This begins initiation, and with the help of other proteins that I'm not labeling here, the large part of the ribosome comes and it sets everything in place. This is the translation initiation complex. Okay, so here you have it in a bigger picture so you can see. You start with the small RNA. It binds to the mRNA binding site, probably the G' cap, the 5' G cap, I'm sorry, and the untranslated region. It looks for that first AUG, the initiator, the first tRNA that can base pair with that AUG comes in, and lo and behold, it's always going to be carrying methionine. Okay. Ooh, the only thing that I hadn't highlighted previously is that once this first part forms, we are going to use some energy. We're going to need some energy in the form of GTP, similar to ATP, right? We've seen GTP before in cellular respiration, at least, or similar to the nucleoside triphosphates. So it's got these energetic triple phosphates that can provide you with free quotes, quote unquote, free energy to catalyze the reaction, which is bringing the whole rest of this ribosome in. So forming this whole complex takes energy. And energy, in this case, is GTP, guanine triphosphate, as opposed to adenine triphosphate. Next step, we got to elongate this protein chain, right? This polypeptide chain, right? And yeah, you can keep drawing it if you want. Did I draw it better here? No, probably didn't. Not so much. But let's see if I can sort of draw it along with what's going on. Oh, this is already near. All right, let's just say that, no, I know it. Let's go back to where I was at right here with my ugly drawing right here. And let's just say that the next one over, notice the the codon, let's just write a bunch of random codons there. The next codon is available for binding. And there's tRNAs all over the place carrying their amino acids that are nearby, right? And only the correct one with the correct anticodon can bind to this next codon, CCC. So it's going to be G, G, G. That's the one that can base pair there. That's the one that's going to sit in there. And that's my tRNA. And you can look at your genetic code. What did I make that? GGG. I'm going to look at my genetic code, look for G, G, G. And this guy I know is carrying the amino acid glycine. Okay. Thanks to that next amino acid, that's right. It's not here. That's right next to it. The ribosome itself. So let's do this in brown. The ribosome itself and its catalytic functions are going to be responsible for forming that peptide bond between these two neighboring amino acids. Ribosome catalyzes, which means it's going to take energy. Peptide bond forming between the neighboring amino acids, and only between those neighbor amino acids. It's only you have a dipeptide. Elongation means, so that's the first part I showed you there, was basically the codon on the mRNA is going to be recognized. The tRNA is going to bind, the correct tRNA will bind base pair, let's say, to the codon, bringing its amino acid, brings amino acid with it. The ribosome catalyzes the peptide bond. And then last but not least, translocation. So I'm going to try to draw it on my translocation is when this ribosome is going to move a codon over. Okay, so let's pretend that I like this stuff at least. It's going to move one codon over. Oh, I erased some stuff. And now it's going to have, I'm going to draw the whole thing together. Now these guys are going to be, this new codon is going to be in the A site. You're in the P site and you're in the E site. We're going to be ready to dump this guy out. So we're going to get rid of the tRNA in the E site. The methionine is going to stay behind in the P site because that's where the polypeptide is growing. And now you have No, come on. Where's my ribosome? And now you have this new codon available for binding. It's only going to bind to what can complementary base pair to the CAG. Well, it's going to be a GUC. You can find out. You look at your genetic code, you look for the codon CAG, and I know that this tRNA must be holding the amino acid. What is it? CAG? Glutamine. Okay, and so this is going to keep happening. Everyone's going to move over a codon. You're going to translocate, move over a codon until you finally have, get them out here, you have this growing polypeptide. Look at, this is going to be this step right here. I'm going to call it A, where this tRNA is going to recognize this codon. Your ribosome is going to catalyze this formation of a peptide bond between the neighboring amino acids, remember, between neighbors only. That is going to require energy. And then when the ribosome is going to move over, translocation, so that's B. C, when the ribosome moves over, C, that's also going to require energy. But notice that's going to let you start over and a new tRNA can come here and base pair. Last step, termination. You're going to stop, right? Happens when you reach a stop codon in the mRNA. So there's 3 codons that label stop translation: UAA, UAG, and UGA. Remember, you'll always get a genetic code, so don't worry about memorizing these, with the exception of that first AUG, which is for start. Okay, so notice you've got your EPA sites on your ribosome, you've got your growing polypeptide in the P site. In the A site comes something called, this protein called a release factor. And it's the one that binds to one of those 3 stop codons. You can bind to it. Instead of it adding an amino acid, it actually causes a hydrolysis. It adds some water to unbind your tRNA from the polypeptide. Let's see, hydrolysis to remove the polypeptide from its tRNA, which means this whole complex, okay, your free polypeptide is going to go to wherever it needs to go, to the membrane. It's going to be secreted outside the cell. It's going to go to the Calvin cycle, wherever it's going to be needed, right? And then the ribosome will dissociate. It will fall apart. These steps do require energy, but that does stop translation. Okay, your polypeptide's made, and remember, you could have been making this the ribosome could have been making this just in a ribosome that's in the cytosol. And that means you're going to use these proteins in the cytosol. So it's going to be used for, if you're a plant, it's going to be used for, I'm sorry, if you're doing cellular respiration, it's going to be used for the glycolysis step. Or it's going to be used for, something guiding microtubules along vesicles along microtubules. If you've got bound ribosomes, that means they are, remember, ribosomes on the rough ER. That's what the R stands for, ribosomes. Well, I like to think about it as it's actually rough ER, but ribosomes make the rough ER rough. Little circle. And any of the proteins that were made on your RER are going to be used either, they're going to be transported to the membrane. They're going to leave, they're going to be secreted out. There might be some sort of signal peptide. They might be used for any membrane though. So nuclear membrane, other membranes, plasma membrane. And that's about it. So just sort of notice that the end part, that one mRNA molecule can have multiple ribosomes on it. That's going to help all of us make proteins more quickly. Polypeptides. We forgot to mention one more thing about targeting. Even before you're making your polypeptide, they're going to make sure that they're going to fold. The polypeptide is going to fold into its secondary structure, its tertiary structure, its quaternary structure. If it's got lots of polypeptides together, make sure it functions well, and then it may be secreted out and may go to the membrane, may go be used in the cytosol if it works. If it doesn't work, it may be targeted for degradation. That's about it. So go ahead and check out any of these videos. And I think I stopped it somewhere right there. And I'll give you some practice problems in class.